Chapter 15, question 9, asks you to describe the structure of a lipid bilayer. Now we saw in question 18 what the lipid bilayer does. It separates the inside and outside of the cell by not letting those polar mixtures mix with each other, by providing a non-polar barrier. But what does it actually look like if you zoom in to here, the orange line, that lipid layer? So that layer is made up of the lipid that it's made up of are phospholipids. And we talked about this in an earlier question. And this is the structure of phospholipid. You have the glycerol backbone, two fatty acids. This whole portion is non-polar. And then you have a head, a phosphate group, the PO4, lots of oxygens there, so it's really polar. And then an amino alcohol. An amine has a nitrogen, and an alcohol has an oxygen, and so that also is really polar. So that part of the molecule is very polar. So you have these two parts, the polar part and the nonpolar part. And what you will do for this is, you take this phospholipid, and if you take these and just put them into water and shake them up, then you will get, they'll just spontaneously orient themselves like so. Now we're going to have one layer of them like this. This is one layer of phospholipids. And what they're going to do is you're going to have water on the outside of the cell. So the polar part of the molecule, the blue part of the molecule here, is all going to face the water. Okay. So that's one layer. That would be a lipid monolayer. But if this were all, this would not be stable because you would have water on the inside, and that would not mix with the nonpolar tails here. So there's another layer, and in the second layer, you take these phospholipids again, and they turn, and the nonpolar tails all face each other. So now the nonpolar parts are all facing nonpolar things, and the polar parts are all facing polar things. So you'd have water inside the cell. So that is polar inside the cell. And you'd have water, which is polar, outside the cell. And what you would want to notice, and the features you want to notice, are these polar heads of phospholipids. Polar heads are made up of PO4 and an amino alcohol. And then you'd also want to notice the nonpolar tails. Of the phospholipids. Those are the fatty acid tails that we saw up here. Notice that the nonpolar tails face each other. And that the polar heads face water. And so that's how these form a bilayer. The two layers, you have one layer of phospholipids, a second layer of phospholipids, two. So that's two layers, that's a bilayer. Bilayer. And when you're drawing this on your sheets, you wouldn't have to draw this whole thing out. It would be nice if you could draw a phospholipid up here, but instead of drawing all a million of them to the side, you can just draw a circle to represent the polar heads and two squiggly tails on each one to represent the nonpolar tails. And that's often how they're represented in biology books and anatomy and physiology books too. So two layers of those with each of these parts outlined is a complete description of the structure of a lipid bilayer.